Okay, Auzubillahi minash shaitanir rajim. Bismillahirrahmanirrahim. So today we are going to discuss about RNAi technology, which is also known as RNA interference technology or siRNA molecules. So short interfering or short interference or even small interfering or small interference RNA molecules. This is the online lecture number 13, and we are going to have the interactive session tomorrow on 13th May. And this is the second last lecture of health biotechnology chapter. Let's discuss about it. So Rosalind Lee was the first person who was studying on C. elegans, which is a nematode, and she was discussing some gene regulation, is LIN4 and LIN14 genes, and there was a silencing effect. We know in genetics, one gene can be silenced in such a way that it does not express, it does not produces its proteins. It could be at the DNA level or at the RNA level. Nonetheless, so she was working at C. elegans and the gene silencing of LIN14, and there was another gene, LIN4, that was involved in it. And then she found out that there were two small transcripts of LIN4 with roughly in 22 to 61, 22 and 61 nucleotides, and they were found to be complementary to the 3 untranslated region of LIN14. So they had base complementarity, and these proteins, these two transcripts, they were not encoding any protein. And then from this, she deduced that these transcripts are the causes of silencing and causing the RNA interactions. The world has to wait 7 years to find out about these kind of RNAs, as the second small RNA was discovered in 2000, 7 years after. These small RNA molecules, they are known as non-coding RNA molecules. Why they are small? Because they are short in length as compared to messenger RNA molecules. Secondly, they are called non-coding because they do not encode any proteins. They are RNA molecules, they are short, and they do not code any proteins. So we call them small non-coding RNAs, or only non-coding, or only small RNA molecules. There are actually 3 types of small non-coding RNA molecules. 2 are very well known, which are known as microRNA molecules and miRNA, and short interfering RNA molecules, which are known as siRNA. The third type is shRNA, which is short hairpin RNA molecules. They are similar, but way beyond the scope of your course. So now we see the protein coding RNA molecules are the one that are in the green, and then there are non-coding RNA. So the non-coding RNA, we talk about short non-coding RNA, and we are not talking about long non-coding RNA. And you can see in the structure, they are making these hairpin loops, and because of base complementarity, they can make these kind of hairpin structures. And when you say RNA molecules are single-stranded, think, because this is a double-stranded hairpin loop. Well, it's not like they're not 2 strands, but it is still considered to be a double-stranded RNA molecule. So these small non-coding siRNA are involved in silencing other messenger RNA transcripts. How does that happen? We will come to see in the next few slides. Why do we call them slides? Because they are really short, 20, 21, 24 to 26 nucleotides long. And they are actually synthesized. The precursor form in which they are synthesized is 2 to 3 times longer than it, roughly 61 nucleotides. And then they are cut by a number of enzymes in the cell. And due to this cutting and chopping, the eventually effective molecule that performs its silencing function is 21 to 24 nucleotides. And their mode of action is they silence a messenger RNA molecule by base pairing with it. So if you have a molecule, a messenger RNA molecule, let's suppose you have a messenger RNA molecule. This is a messenger RNA molecule. It has information for, let's suppose, 25 amino acids. Now you have a short 21 to 24 nucleotides siRNA, and this siRNA has base complementarity to this part with some nucleotides. Now what will happen when the ribosome, it wants to translate this from this direction, it can translate here, 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 but not here, because now here there is not a single-stranded messenger RNA, but it is a double-stranded, it's a double-stranded RNA molecule. So the ribosome, theoretically, it cannot translate it. But the story does not reach this far, because when you have another small piece of RNA molecule binding to the messenger RNA, it is chopped off. But nonetheless, when you have this double-stranded structure, it will eventually silence. Silence means you prohibit the translation, you prohibit the gene expression. Of course, if you prohibit, if you inhibit, if you stop the translation or the transcription, that gene will be silenced. But in this case, silence means you're not stopping the gene expression. You're not stopping the RNA transcription. Transcription is carrying on. What you do the regulation at the messenger RNA level when the messenger RNA will be converted to a protein or a polypeptide molecule. There you stop it, you control it, you regulate it, and that's how we call it gene silencing. So it was found in, as discussed earlier, I think we have discussed all these points, that double-stranded RNA molecules was 10 times more effective. Okay, it was found that these are 10 times more effective than sense and antisense. Sense and antisense therapy, I will let you see them in the next few slides, but this is again another phenomena for gene silencing. You have messenger RNA molecules that are sense, and then you have other RNA molecules, they are antisense, which means they are complementary to each other, and when they bind to each other, eventually you will have gene silenced. And the miRNA and siRNA and shRNA, these three types of RNA which we discussed here, the term we just uttered it, we haven't mentioned it here in the writing. They are actually dependent on two classes of proteins, enzymes in the cell. One is known as dicer and the other one is known as argonaute. Argonaute and dicer both have a number of other accessory proteins to them. And there's a Nobel Prize awarded in 2006 in medicine for the discovery of this RNAi technology tool. Andrew, who was from Stanford University, and Craig Mello from University of Massachusetts. Both of them are in the United States of America. Okay, now coming to the basics of genetics. This is the gene in the correct orientation. And when this gene will be transcribed, I mean, when transcription happens, you will have a messenger RNA. Now, what will happen if we change the direction of transcription and we transcribe it in the reverse direction? We will have a messenger RNA, and this messenger RNA would be an exact copy of this messenger RNA, because this DNA strand is an exact copy of this DNA. Same wise, this messenger RNA is going to be an exact copy of this part of DNA, and this messenger RNA is going to be a copy of this DNA. Now, let's recall it. These are two DNA strands, right? So these two DNA strands in their intact form should be a duplex, and their duplex totally relies on the base complementarity. So if this RNA is from one strand and this RNA is from the other strand, it's pretty logical to understand that these two RNA will have to be complementary to each other. In this case, these both gene constructs, not construct, but these messenger RNA molecules, they are full length. So the number of amino acids, not amino acid, the number of base pairs in this messenger RNA should be theoretically the same to this, because it's the same gene. It's the same gene transcribed in two different directions. Now, when these two messenger RNAs, they incubate, or they're incubated together in the lab, under lab conditions or under natural conditions, they would make a perfect messenger RNA sense and antisense copy messenger RNA. And whenever there is a double-stranded messenger RNA or double-stranded RNA molecule in the cell, there are specific cellular ribonuclease enzymes that recognize them, and they will chop it, and eventually what will happen? Gene silencing will happen. Okay, now this is again a sense and RNA antisense therapy. This is a eukaryotic gene which is transcribed. This is the primary transcript. This is exon 1 and this is exon 2, and eventually the RNA splicing. These two messenger RNAs are put together in the form of a mature messenger RNA. Then you have a plasmid, and on the plasmid you have cDNA that is derived from this messenger RNA, and you clone it in this plasmid, and then what happens? You make these messenger RNAs, and this messenger RNAs, they would go and directly attach to these antisense RNA will attach to the transcripts, to the mature messenger RNA transcripts, and eventually they will be having a complete base complementarity, and it will let the messenger RNA not to be translated. Eventually, gene silencing will happen. So why we put it on here on a plasmid? This means that you can actually clone this gene construct into the genome of this particular organism, and you do not need to provide these antisense messenger RNA as an extra dose. So when this gene is transcribed, this will be transcribed, and eventually this gene will always be shut down, or it will always be silenced. This is one of the ways we can do gene therapy. When you have a gene therapy and you insert this gene, you want this gene to be silenced. If the product of this gene is damaging the body, you can have gene therapy, design a viral vector, have the cDNA of this messenger RNA on this plasmid, or just viral vector. Once it's integrated into the genome, when this messenger RNA is made, there will be the antisense RNA molecules already present, and the duplex of them will make sure the gene is silenced. Okay, in this case, it's the same story. It's 100% the same story. The only difference here is you are providing these antisense RNA molecules in a short form. You see, now this is not covering the whole gene. In this case, we were covering the whole gene, but in this case, it's not covering the whole gene, just a few nucleotides. This is actually what is the non-coding small interference or miRNA molecules, and from studies and from experimental evidence, it is proven that this type of gene silencing, in which you use shorter nucleotides of antisense RNA molecules, they are more effective than the full-length gene constructs of antisense messenger RNA molecules.